I'm the President of the Royal Society of Edinburgh, and John Arbuthnot. It's a pleasure to welcome you to this particular lecture, which is a lecture supported by the Scottish Cruden Foundation, and we're very indebted to the Scottish Cruden Foundation for continuing support for this lecture. And the Scottish Cancer Foundation itself has been much involved in organising this meeting, and the relationship between the two foundations is very important in taking cancer work forward in Scotland. So let's welcome our speaker, Andrew Biankin, who's Director of Wolfson Cancer Research Centre and Regius Chair of Surgery in the University of Glasgow. His topic is going to be about cancer genomes and cancer care. And for those of us who are not particularly expert in cancer, I just want to set the scene a little bit. And Andrew will take us through from there to some extremely interesting work that's going on both clinically and in research. I think what he and I would like to get across at the very beginning is that there is now beginning to be a different philosophy about the pattern of the development of diseases, including cancer, in individuals. It's quite difficult to change the philosophy that cancer is cancer, liver cancer is liver cancer, pancreatic cancer is. But the way in which individuals react to the initial stages of that disease can be very significantly affected by the way in which their genetic makeup works. And that, to some extent, can be inherited or something that happens at an early stage in life. And I think Andrew is particularly keen to get that message across. Do you agree, Andrew? So that then leads on to the idea that instead of just treating everybody who has a particular disease in the same way, you have to personalize that. In order to personalize it, you have to know much more about the makeup of the person. And the way in which that is done nowadays is to use the fantastic advances in molecular biology to do it, and it can be done really very speedily. And Glasgow is a centre of that personalised approach, and they are developing a whole programme of what we call stratified medicine, so that the way in which we stratify the treatments of individuals is different from that that's happened in the past, and much more directed to the personal circumstances of the individual. So I think that's as much as I would want to say, Andrew, about the introduction, and we look very much forward to your talk. Thank you very much.
Thank you very much, and thank you for the very kind invitation and the opportunity to address you here this evening. Really about what the President was saying, and that was about personalising care. We made decisions in the clinic based on what we observed with our eyes initially. Then we had a microscope, and the microscope is a 300-year-old technology. Now we have technology that allows us to peer into what makes a disease a particular type of disease. Not only does it start to break down what we originally thought was one and the same thing, it's breaking it down into multiple different parts. And it's little wonder why one person will respond to a certain treatment and another person won't. And currently our public health systems really group all of these things together. Whereas we need to change the way we're approaching disease in general, and in particular cancer, because the technology we currently have focuses on understanding genes, and cancer is a disease of genes. So we're starting to really focus down on that. And what brought this to my attention very early on, I had just finished my surgical training. I'd started my PhD and started my practice as a consultant. One of my first patients was a 39-year-old woman with a tiny pancreatic cancer. It was maybe a centimetre at most. And I just finished my training. I was about as good as I was ever going to get, and I did an operation that I thought I was really proud of. This is fantastic. And she sailed through it. Of course she sailed through it. She's 39 years old and very fit and healthy. I saw her about 6 weeks afterward. Everything was going great. She was fine. I said, well, you know, the pathology came back. It hadn't spread. It was a tiny little tumour. And I thought, wow, okay, this is going to be great. 10 months down the track, I get a phone call from the hospital I was working at saying, your patient's come in and it looks like as if his cancer's come back. And this was a tiny, small thing and clearly indicated to me we really don't know what we're dealing with here. We have a rough idea and we really get a few opportunities that we take forward, but we're really almost stuck in the way we manage disease because we're trying to treat everybody with the same thing. And we've known for 1,000 years that when you're starting to treat a particular disease or understanding a particular disease, you should make sure that it is the same disease you're testing your drug with. We wouldn't dream of giving a treatment to cancer just as cancer. We say it's breast cancer, it's pancreatic cancer, it's prostate cancer. When you have a lung infection, you try and understand what that infection is. You don't just say, well, it's a lung infection. And this is just the next way forward of really understanding differences between people and the diseases they develop. And it applies across the board, not just cancer. Your risk for disease, your risk for cancer, your risk for developing any sort of disease. And starting to understand that and how we manage that in the future is one of our challenges. We're starting to be able to do the science, but the problem is we're stuck in our old ways and we need to develop new ones to be able to deal with these new data and these new information. And our regulatory authorities are driving this to a certain degree, and so there's a lot of complexity that needs to change. So what is cancer? So cancer is essentially cells growing out of control. But it's not just that, it's cells evolving. So as we all evolve as individuals or species, etc., over time, similarly, each individual cell wants to evolve. And it's an evolutionary pressure, and it's basically how bacteria evolve, how anything evolves. And these cells are trying to escape the confines of their environment and basically live as individuals. I wasn't going to say the independence thing, but anyway, they want to live independently. So everybody wants to be independent. And this is about really breaking through some of these barriers that cells have to being independent in this particular state. And this can happen either through the slow accumulation of genetic damage, or sometimes it can happen with a sudden catastrophe. And these are the things that we study and try and understand, these particular defects in the genes, some of which we're born with, and others that we develop over time, either through different environmental exposures or through other processes, which I'll touch on. So what's driving this is advances in technology. And I'm sure some of you would be familiar with Moore's law. Moore's law applies to computer chips. It means every 6 months, basically, it halves in size, doubles in speed, and halves in price as well. Well, the advancement of genomics is exponentially faster than Moore's law. And what it allows us to do is to really understand every single bit of damage that can occur to our genes and to map those together and understand those particular elements and what effect they have on the evolution of cancer. So we started this a few years ago, and just to give you an idea of how technology has enabled us to do this, it's taken genomic technology but also computing. And so the amount of data we now generate, we generated over a 2-year period about 4 petabytes of data in Sydney. We wanted to move that across to Scotland, and it would take 2.5 years on internet, on your fastest internet connection, to move that across. So we have to buy a server and shift it. And at the moment, this is a picture by somebody called Eric Green, who's the head of the National NHGRI, Genome Research Institute in the United States. And so what this demonstrates is where we are from the top left-hand corner, really just understanding the structure of genes, the structure of the genomes, and then how it's starting to move forward through patient care. And these heat maps, the redder they are, is there a laser pointer here? It might be. No. Anyway, the red spot means there's a lot of activity. So we can see we started off basically in the top left-hand corner, just understanding the structure. But as time progresses, we get more and more movement into the clinic. How is this going to affect healthcare? And it's a tidal wave coming. How do our systems deal with this? When the technology really got to the point where, by comparison, the human genome was sequenced in 15 years and $3 billion to sequence one person, and that was basically in 2001. In 2009, we could sequence basically one normal genome in about 3 days at the cost of maybe 50,000. Now we can sequence a normal genome for less than $1,000 in probably around about 18 hours. So this is really driving how things change. So when we saw this coming, Tom Hudson from Toronto and many other funding bodies met to form the International Cancer Genome Consortium. This is really 24 countries coming together and saying, what are we going to do with cancer? We've got all this technology. We really need to understand cancer better. So various agreements were made as to how we start to map these cancers. And it's really kept mapping out 500 of each cancer type. And you can see various contributions from different countries, a number of contributions from the United Kingdom. And we worked on pancreatic cancer, which we have now, in fact, Scotland is part of the International Cancer Genome Consortium. Sort of officially, because whenever they put me on the program, they put me down to Scotland, and this is Scotland's representative. So we, by de facto, haven't got any money from the Scottish government yet. But so pancreatic cancer, it's the 4th leading cause of cancer death. Less than 5% are alive at 5 years, and 90% die within a year. It's a terrible disease. Surgery works some of the time in a small proportion of patients. And my oncology colleagues use this term modestly effective, which means it doesn't do very much at all, but it's nice to say that, nicer to say that. And nothing's changed in 50 years. And so I started work on pancreatic cancer for a number of different reasons, but we also see this as an opportunity where we can start to understand how we can implement this personalized approach to cancer. And why we think, because we don't have a personalized approach to cancer, why we're not really advancing things for pancreatic cancer. And so what we did in our study, we recruited 500 people who had pancreatic cancer, and we tracked them like a clinical trial. When they had their operation, we took that sample and we took it and we sequenced it, basically looking at all the genetic abnormalities that we see inside that cancer. And I'm going to take you through some of those in a little bit of detail just to portray how we start to take this into the clinic. And when we did our sequences, what we saw, and this is what's called a word cloud, and the size of the letters represents the frequency or the prevalence of that particular mutation. Each one of those represents a different gene. And that doesn't say CRAS. I didn't realize that. I just always knew that was as KRAS. But the first time I presented this in a public audience, I said, why have you got CRAS on your screen? You can't really do that. And some of the gene names can be quite crazy. So what we saw is 4 genetic abnormalities we'd known about for a long time. But what we saw else was this remarkable complexity and heterogeneity. And we thought, little wonder different patients have different cancers that act differently, that respond differently to treatment. But what was important is that in pancreatic cancer, we did not find a particular subgroup that had a drug towards it that was greater than a few percentage points. So we have tried all these drugs, we tested them, but because only 4 or 5 patients out of 100 responded, it was below the noise level of the clinical trial and we could never detect those differences. Others where we've made dramatic advancement in personalized therapy, and that's really anti-estrogen therapy or tamoxifen for breast cancer. And that's because the proportion of patients with that target for that drug is about 50 or 60%. So our systems aren't really geared to understand this or to test this or to really face this in drug development. And so this has significant clinical implications. We also use other techniques to help us try and understand what's an important genetic event, because sometimes genetic events contribute to the cancers, but sometimes they're passengers, and you don't want to spend a lifetime making a drug for a particular genetic event that isn't going to make any difference. And we use different model systems to inform the human disease. Here we can use screens, etc., manipulating cells in order to understand more about a particular genetic event. Then use animal models to understand those relative differences. Again, other different animal models to really look orthogonally, compare species that develop cancer or that we give cancer to be able to work out what's important, what isn't important. And that resolves us down to particular pathways or processes that we can start thinking about this complete mass or morass of different genetic mutations into pathways where we can start to think about how we develop drugs or we particularly target them. So there are 12 original pathways that were described. We added a couple more to those, and so they were studying them in more detail. So this is what a whole cancer genome looks like. So your DNA is composed of 22 chromosomes plus the 23rd, which is your sex chromosomes. And in that circle, which is called a Circos plot, the chromosomes are arranged around from number 1 all the way through to the sex chromosomes. And we can start to mark out where these genetic defects are occurring. So, for example, around here we have a type of genetic defect called a mutation, which is just a change in one base, and there's 3 billion bases in the genome. Whereas here, in the middle, these massive lines are big bits of DNA that float off from one spot to another. And they damage lots and lots of genes, and hence identifying which are driving that cancer, which are important to that cancer survival, is vitally important and an area of a lot of research. When we start to try and map out these particular cancers and we think about them in pathways, when we start to see groups of mutations emerging, then we can start to think about where do we target these drugs? What is it about this pathway? Is the pathway increased? Is the gene more active? Do we need to make a drug towards that gene? Or is it representative of something different? Looking deeply into these genomes of cancers, which is the cancer versus the normal, we start to see patterns of biochemical patterns of the base changes themselves. And so these are called signatures. And what they tell us about the cancer is the very core biochemical processes that cause that cancer to develop. For example, exposure to smoking. So smoking leaves a particular signature in your DNA that you can see in a tumor to say that these are the mechanisms that were operational that drove that cancer in the first place. Older age has a certain particular pattern. We can see inherited mutations like BRCA1, BRCA2 that are inherited and can predispose to cancer, but they cause a particular signature of genomic scarring that pushes the cancer to or increases the risk of a cancer developing. There's an ultraviolet light signature where you get in skin cancer, and many others as well. And these help us understand what might be driving that, and has particular implications for screening, early diagnosis, and particularly for prevention. And this slide I just brought up just to say that that's one element, but you also see other components that act upon the genome, the methylome and other bits. And across the bottom we have the patients, and across the top we have all of these different events. So making sense of those events and having the computational power to be able to analyze those things is significant. It's a little better now because the computing is getting better, but when we first started, you would basically press start on your computer and you'd walk away for a month. And it was a computer that was equivalent to 3,000 laptops, and they'd just be churning through the numbers. Now we can do that a lot quicker, thankfully, and that's why we're thinking about how we can put this into the clinic. And so I'm going to talk about precision therapy, but I'm going to talk specifically about pancreatic cancer and use real-world, real-life examples as part of our experience as we did this work. So think about this, our old classification of cancer. We had breast cancer, colorectal cancer, NSCLC is non-small cell lung cancer, prostate cancer, melanoma, a whole pile of others, and GIST. But because we can now resolve differences at a molecular level, we're starting to see all those different colours, different subgroups, and those subgroups have clinically relevant information, and they allow us to select the particular patient for a particular drug. And that's really the premise of personalised medicine, the right drug for the right patient at the right time, at the right cost, and in the right outcome. And this has significant economic implications as well for healthcare, in that if you have a subtype that's only present in 5 or 10% that was responding to a particular drug, you're giving 95% of patients a drug that isn't going to be helpful. Not only do they miss out on opportunity of getting the right treatment first, they also have a risk of toxicity, and also it diminishes the overall healthcare budget so that we're wasting therapeutic opportunities. And this I'd mentioned previously is if it's so diverse and there are so many differences that we see, we can't really be testing the same treatment strategy. And this is the second rule of clinical trials, and it's almost 1,000 years we've known that, and yet we still persist. We know that these are different cancers, and we still try and do the same therapies. And so we need to change that. So one of the particular challenges we have in pancreatic cancer is that most patients don't receive treatment. This is the sort of treatment that they receive. This group doesn't get any treatment because they're unwell, and they only survive 2 months. And at the moment, we've made some advances in pancreatic cancer, but they're given a drug called FOLFIRINOX, which is a combination of 3 or 4 different drugs. And you can imagine it's quite toxic. So you're really going for the big hit there. You're going, well, I'm going to take this drug. I've got a 5% chance of it working, and that's the best chance I've got. 95% chance it's not going to work, and my life's going to be miserable for the last period of time, which is going to be an average of 10 or 11 months. And what we're really trying to do is unlock that and say, well, we want to pick the right drug for the right patients for the treatments we have, and then give the opportunity to the other patients that aren't going to respond opportunity for novel therapies. And it also allows us to find new treatments as well on top of that. And when you look deeply into these cancer genomes, you actually see opportunities for treatment right there and then with drugs that are already available. We've tested them, but we just haven't given them to the right patients. Drugs that are, definitely, even Herceptin used in breast cancer, there's an opportunity for Herceptin because we see exactly the same genetic mishap in a pancreatic cancer, in a gastric cancer, in a stomach cancer or colon cancer, and in a breast cancer. We just happen to know about it in breast cancer because it's present in about 20% and we've detected it. But we see that same amount there in the same mutation in pancreatic cancer, but it's only present in 2 or 3%. And so that's, how do we pick those patients ahead of time? Because we don't currently do that. When we saw some of these mutations, we had the opportunity to look back into our data and see if by chance the patient got the right treatment. We couldn't really tell them what to take, but we could say, well, just by luck, did the doc give them the right treatment? And in Australia has quite a booming private practice, and docs like to go off-piste a bit. So we could look back and see what happened to these patients. And so when we got it right just by accident. We saw patients having dramatic and exceptional responses, things that we'd almost never seen in pancreatic cancer before. At the time of recurrence, this is a particular patient. This line just represents a tumour marker, which is called CA19-9, which tells you the amount of disease there is present. And so at the peak of this point here, this is the patient's scan. That's a liver. That's just a cyst, James, just in case you're thinking it's a metastasis. And that's the recurrent tumour there. She went on, just through chance, on a treatment called Folfox, which had a platinum containing it, and the tumour completely went away. The portal vein went back to normal. The blood level went back completely to normal, and 4 years down the track, she's still alive. Almost never see that. But when we look back, we could start to understand or develop hypotheses as to why that was the case. There's another example of a patient who got a different therapeutic, another metastasis in the liver. But when we took that metastasis out, because the patient was sort of off treatment and doing well a couple of months down the track, and we thought, well, what do we do with this thing in the liver? We better take it out. And we took it out, and it was completely resolved. It was a cancer that had complete pathological resolution. We see different patterns associated with particular cancer subtypes. So when you think of pancreatic cancer, you think of mostly the recurrence is going to be in the liver, but there's a subgroup where it doesn't recur in the liver, it recurs in the lung. Completely changes the way we should approach this. So rather than doing it in a broad way, what's right for 98% of patients, we start to dissect out what's going to be an important aspect for the patient in front of you, for that particular individual. So we can do this in small groups. Lucky individuals, sometimes we do it for high net worth individuals because we don't really have a choice. But how do we test these when we've got small numbers of patients? Even through the UK, some of these might only have 40 or 50 patients a year. If you tested everybody, and this is what just power calculations to predict the size of a clinical trial, if you were lucky and 10% of your patients had a genetic defect and you had a drug for that particular defect, in order for you to detect a difference, you would need 7,540 patients. The largest pancreatic cancer study we've had in advanced disease has been 800 patients. So you can see why we can never detect that. It's really got to start working in 30 or 40% or better. And so we're really starting to do clinical trials very differently now. We identify those patients with the right particular subtype of tumour, and then we group them together and we give them that treatment, and we look for bigger effects in that particular subgroup. That's changing the way we treat cancer, we treat other diseases. But our processes are built like the top one there. It's a serial model. We only do a test when we think we're going to respond to that test. And so you have a treatment, you have a treatment, you have a treatment, and oh, we need to do a test now, oh, we need to do a test now. Because we can now deliver these diagnostic tests, we can now do them right up front, and it develops a parallel model. So if you're an individual with a cancer that says, well, I've gone, and I get this all the time now, patients will send their sample to Foundation Medicine in Boston, and they'll say, I've had a Foundation 1 test, this is my cancer genome, doc, what do I do? And it's a challenging position because there might be standard treatments available, but the individual says, well, I've got this thing, and there's a drug for this thing. Why can't I get this thing? And it has all sorts of challenges across the board, from an ethic perspective, from a management perspective, etc. But what it does give us in pancreatic cancer is that we don't get a lot of patients on clinical trials because we're trying to shoehorn a particular patient into a clinical trial. We've got one trial open. This is a great trial event. We designed this, but it's only suitable for a number of patients, not all of them. So this gives us the opportunity to turn that completely around, and rather than finding a patient for the trial, to find a trial for the patient. So completely different way of thinking. So when you arrive as an individual, you don't have the standard of care or one potential clinical trial. There's a multitude of options that you can choose from, and you can decide which one's going to be best for you and what suits you best. And in order to do that, we're developing systems where we test everybody's cancer at the time of diagnosis. We identify all those different subgroups, and they're all different colours just to represent a different subgroup of cancer. Then we match them in parallel to different treatments, so each patient gets an optimal treatment straight up front, and they get a much better response. And then we can continue to develop this in a clinical trial in a research setting. And this is something we're building across the UK, just starting to pull it together with Cancer Research UK and others, and it's called Precision Panc, and it's really about transforming the treatment paradigm for people with pancreatic cancer. Pancreatic cancer, because pretty much nothing else works. We don't have standard therapies. And so we have the opportunity to develop and understand this parallel model of personalised healthcare for pancreatic cancer, where basically nothing has changed for 50 years. And the reason why we think it hasn't changed is because we really haven't understood that diversity and dealt with it, and this is what we aim to do in this. It's not just about having the clinical trial in place, but it's all those elements that come together to make us understand what specific therapy is right for that individual patient. So at a stage towards the end of our project for ICGC, we had scientists just working in our lab 24/7, people belting down the door wanting to work, because they were starting to realise what they were doing in the lab could potentially in 2 months' time have a particular impact on a patient, because we'd opened a clinical trial that said if you have a particular mutation, then we can offer you a specific drug which is right for you. And then suddenly the researchers who had never seen a patient before were thinking, well, what I do today might make a difference in 2 months' time, not in 30 years' time, but in 2 months' time. And then really building those systems where you can discover a new treatment, And this area of preclinical development and there's the so-called valley of death in drug development is really being precise about how we make those systems work and then directly trying to get those treatments to patients as rapidly as possible. But not large-scale trials of thousands of patients, but more specifically looking at small groups of patients to identify which treatment is really going to be the right one for them. And these clinical trials can be complex, but that complexity is our problem, it's not the patient's problem. And we try and identify a treatment for an individual, at least an option to say, well, you can have standard of care, of course you can have that, but look at these other options we have on offer. So everybody has the opportunity to start to get the optimal care. A big challenge is really to educate the healthcare community. You would get a report, this is your potassium, it's just one number. It's pretty easy to interpret. Then you get faced with this. How do we train our community to understand this? To say, well, this is your genomic report. We have a wonderful opportunity in the UK to do that because of the unified health system. The US have been trying to push this out for the last several years and they can't manage it because it's so fragmented and it's a completely different model. And what this really is starting to build on is a different approach to healthcare overall, which is a knowledge bank approach. So if we have these subgroups, if we're all unique or we have fairly small subgroups of disease, then how can we do clinical trials in all of them? We just can't. But at the moment, we don't really collect enough information about patients, treatment, their various test results in order to be able to infer that. So over time, what we're trying to build are these knowledge banks and have a rapidly learning health system. So rather than having a clinical trial, you're out of options as an individual patient. Rather than saying, well, we don't have a trial, let's toss a coin or guess what drug we're going to give you, you can say, well, I've looked at your tumour, and your tumour says you've got this particular mutation profile. I'll just look up in the knowledge bank to see if we had anybody similar. Okay, your tumour sort of matches pretty well with another 100 people out there. They all receive these different types of therapies, but 10 patients received this particular type of therapy and it worked really well. So we're going to give you that particular type of therapy. Completely freaks the regulators out. I don't know how we're going to get past that, but we just have to get past that because otherwise we're just going to be, not going to make any significant advances forward. One of the key elements is accumulating those data and bringing those data together. I mean, what's it called again? Google Glass. That's right. It's called Google Glass. And so what it is basically is an iPhone in your eye. So you can just look, you can basically look at that there and you think, what a great advancement. It's going to do great things for chatting to your friends in the middle of the night. But why don't we use this in healthcare? Can you imagine when I was an intern in the emergency department, somebody, it's 3 o'clock in the morning, you're sitting there barely staying awake and suddenly everything goes crazy because the ambulance comes in comes a patient. What's wrong with them? We don't know. What's wrong with you, ma'am? I don't know. What drugs do you take? The little blue ones. Whoa, okay, so you're sort of working, you call for the old notes and you're waiting, and you're working and suddenly she arrests and dies and, it's not very good. And then 2 hours later, bang, this whole pile of old notes arrives. And you think, gosh, if only I knew this, maybe I could have made something different. And even if it came there and sat there, you think, oh, excuse me, just hold on there for a second, I just want to read through your old notes. Could you imagine if while you're sitting there in the emergency department, the ambulance just clicks a button, bang, up comes on your screen a complete history of that individual patient, knowing what they've got. I mean, this privacy issue is, sure, it's important, but I'm the reverse. I want every doc in the world to know what's wrong with me. So even if they just see me, they can say, right, okay, this is what you need instantaneously. It's an opportunity to get the better healthcare. We always have to be concerned about misuse of these new technologies. So watching the football on a first date probably isn't an ideal thing to do, but maybe worth testing on occasion. And look at this. This is a contact lens that real-time monitors blood glucose, your sugar content. Reports it to your iPhone, and your iPhone goes beep if it's up or down, right? Okay, so we can do this. So, what are we going to do with those information? The opportunity for research is stupendous. We're also trying to develop things for cancer that rather than taking a blood sample, and some tests are very difficult. You need quite a bit of blood, and you can't really take 5 litres of blood to do a test. It's not going to really work very well. So we're really putting in real-time biosensors with monitoring, etc. Really trying to imagine how we might be able to take things further. So I'm just going to finish there at that point, and we can have a bit of a discussion, answer some questions and ask some questions. I didn't really dwell too much on the ethical component of the debate, because there's a lot of interest out there and a lot of discussion, and I can get myself into deep water pretty quickly. So, okay. Thank you very much.
Well, I told you at the beginning that medicine was changing, and Andrew has certainly emphasised that. And I'm sure there's a number of things that people would like to explore with you. And I think, bearing in mind that this comes as quite a new concept and a new set of approaches to people in the audience, could you just explain a little bit more about the molecular phenotype report, which in itself looks like the annual report to a company on their financial well-being? Except it's on one page when the accountants usually use 55 pages, but it's a very complex looking thing. So, but you have to have that, and you have to be able to understand it and then take and analyse it and then act upon it. So how do you, and that's taken some time to get here, as you explained.
Yeah.
Could you take us just again through how you got there and how it's used and what it tells you and how, you know, not can every hospital use that kind of thing or not? Yeah. How practical is it? So
that's a very important question, because we can do that for individuals, for small groups, or in very subspecialised areas. But how do you scale it? How do you take it out to the whole population for the opportunity to do that? Not every clinician. If you're in a major central hospital, an academic hospital, you might treat lung cancer and look at the subtypes of lung cancer. If you're in the community, in most places in the world, you treat lung cancer, breast cancer, colon cancer, prostate cancer, and then all of a sudden there's 50 different subtypes of all of those things. And so the way we've been approaching that is what we call a molecular MDT, or a molecular multidisciplinary team meeting. So they're previously, at the moment, we'll have a surgeon, a medical oncologist, we'll have a radiologist, and a pathologist. Now we add to the mix, a genomics expert, an informatics expert, to start to discuss those areas and resolve them into something that's going to be meaningful. Some of the experiences we've had is the first time we started to generate these reports is that the docs would say, no, you don't send me that, that's just ridiculous. I'm busy in my clinic, I just want a one-liner, plus or minus. Okay, that's all I need. And so preferably so my secretary can read it and tell me. And then we go, yeah, sure, no problems. And so they would do that. And then all of a sudden they'd say, well, it was negative. You got something else? What else have you got there? I said, well, I thought you just wanted a black and white. He goes, well, if it was positive, it was great. I would have given him that drug. But what else is there? Can you tell us? Can you show us? And so all of a sudden we had people ringing up and saying, well, tell us more. Tell us what's going to happen here, etc. And that was pretty tough from just a resource perspective. All of a sudden we were swamped, and we were thinking, how do we actually scale this and make that happen? So we worked a bit with a company called Life Technologies. This is about 5 or 6 years ago, to try and develop systems to do that. But we tried to that happen in North America, and that didn't really succeed very well because they're all very independent, each institution. But what's happening in the UK, you've got Genomics England working pretty hard to make that happen. And it's really finding that point between simplicity and complexity, but also training clinicians and getting them early so they grow up as part of being, they all use iPads, they all use email, this is just the next iteration of that, and understanding that as you're training.
Yeah. I'm going to open it up. I'm just going to, I mean, I spent some time as the head of microbiology in Nottingham University Hospital, and also in the Royal Infirmary in Glasgow. And the diagnostic tests we had there were nowhere near as complex as this, and perhaps there were a dozen or 15 things that we did routinely. And that in itself took a pretty vast number of technicians to do. Because you have to rotate, they have to be there 24 hours a day, etc. And this is not trivial stuff. Putting that picture together, how much human impact is there in actually gathering that data?
So that's getting, the capacity issue is not one that scales linearly. You need a certain investment. But then after that, it's not so manual. We try and take people out of it as much as we can, not because they're too expensive. That's one thing, but they just generally make mistakes. So we automate everything right from the start to finish. So we just take human error out of this whole analysis. As soon as the sample comes in, it's run by an automated barcode LIM system. So with the scaling of the technology,
so the whole battery of tests is done automatically.
In the pioneering stages, we're still doing a lot of manual, and there's still manual intervention.
Right.
But that manual intervention is not suddenly, if you have to do, double the amount of tests, you don't need double the amount of people.
Right.
With our new technology, we can do 18,000 genomes per year, and we automate that through a computing system. And the biggest challenge is not the computing and the delivering the test. The biggest challenge is getting the sample in the first place. Right. And that's where we fell down mostly in our clinical trial. We could take the sample, we could prepare it, we could sequence it, we could get a result back in 7 days. It just took 6 weeks to get it from the pathology department, because they say, not from my cold, dead hand are you getting this sample, just in case we might need it. We'd even get patients ringing up and said, look, we can't get the sample. And they go, well, just ring, this is the director of the pathology department's number, you give him a call. And even then, and the only thing that succeeded was bribery. So in the end, we just gave them, we said, if you can get the sample to us within 72 hours, we'll give you $200. And it worked like a dream.
Right. Okay. Yes.
Thank you very much for a wonderful lecture, lucid and also humorous. I actually am a pathologist, so you will understand that I'm a little sensitive to some of the comments that you've made. I assume they were referring to your Australian colleagues.
They were Australian colleagues, of course. We have no issues here whatsoever.
I was going to ask you, if I may, the area that concerns me, I spent a considerable time of my life making the decision between cancer and not cancer. Now, from the information you provided today, it's clearly that they're all cancers. And you're absolutely black that they are cancers. But life is not like that. And I wonder if you could spend a moment to speak about the borderlines of making a disease cancer or not cancer.
Yeah, that's a good point.
So there are a couple of issues there, and you're spot on. So we highly selected, the mandate for ICGC, primary cancer, that's what we focused on. But of course, from a pathologist perspective, you're dealing with a whole bunch of different diseases. So what comes to us in that initial aspect is this is cancer. But you're absolutely right. What about precursor lesions? What about the borderline type lesions? What even about classification? So one of the challenges we're seeing is that what is clinically and pathologically one type of cancer, molecularly it's something very different, and we can't resolve, is this an unusual pancreatic cancer, or is this actually a colon cancer? So this concept of KRAS wild type in pancreas, we think every pancreatic cancer, 95% has this particular genetic mutation, but 5% don't. Is this a different diagnosis? And we're about to start to ask those questions, is to take 3,000 cancer genomes, put them into the cloud, and it's taking us about 6 months just to upload them, and ask that analysis and try and resolve the question between the histopathology, the molecular pathology, the classification, the differences between there. And the next step is to build on to How do we distinguish this from precursor lesions? What's the taxonomy, and how, when you're with the clinico-molecular taxonomy, the clinical, etc., and what the relationships are. And it's not an easy question, but we're just starting with a diagnosed cancer and really trying to change the therapeutic aspect of that.
So the shades of grey are still there?
Yes. Oh, yeah, yeah, look at that. It's even greyer. Yeah, the shades of grey are definitely there, and we're really still only just getting the low-hanging fruit. And at least for pancreatic cancer, we see great opportunities for repurposing and rescuing therapies that have been abandoned or used in others because they're targeting a molecular defect. Just because it's a molecular defect in another cancer, we don't know if it works or not. We haven't tested it appropriately. That's what we want to do.
Just following up on that question, I mean, is there a weighting of the importance of, or the relevance of a particular test, albeit that you've got 20, 30, 40, 50 different tests? How do you weight the outcome, or are they all weighted equally?
So that is the current challenge. So you try to differentiate what is a driver mutation, which is actually making the cancer happen, versus a passenger, which means it just happened to occur there, but it's not really making a difference to the cancer in any shape or form. Then what is a particular, Achilles heel of that cancer? What's a therapeutic target? So trying to resolve these issues, and it's challenging because I'll give you one example. So we did a neuroendocrine study, pancreatic neuroendocrine tumour. Everything clinically, histopathologically was a pancreatic neuroendocrine tumour. We sequenced it, and it had a signature of ultraviolet light exposure. So, as far as I know, the pancreas doesn't often see light. Even when I'm standing next to the window operating something in some hospitals, and we scratched our heads, and then we chatted to the pathology. He goes, yeah, it looked a bit funny. Yeah, so there's something there. We don't really delve into that precision as to even the clinical stuff. We're just still bunching it a bit too much together. And we've done it in leukemias and other diseases by being taking careful attention. But in pancreas, we just haven't done it. And so anyway, we go back to the patient and go, have you had any other things going wrong? And he goes, oh, yeah, I had this thing cut off my shoulder. So we looked at it. Yeah, you've been mouthing what it is. It's a Merkel cell carcinoma, a metastasis to the thing. And you think, whoa, that's just crazy. That we never thought that could ever happen. But yeah, we'll learn a lot more.
Right. Next question. Who? Yes.
Obviously, the advances in molecular signatures have been fast and no doubt will continue. I'm not a pathologist, I'm a microbiologist, so I don't actually like having the tissue, or don't have tissue so much. But what do you think is the future of liquid biopsies, i.e. serum or plasma, to look at the same kind of signature and still use it for diagnostic and treatment options in a personalised way?
Agree. I mean, that is the holy grail to a certain degree. That's where we want to get to. We're still a long way away from doing that for a number of reasons. We don't. One is a sensitivity issue, and this is one I think I was talking about. You need 5 litres of blood. If you want to try and get an early detection marker, and it's coming from a precursor lesion, particularly if it's intra-abdominal before it passes to the liver, it's hard to detect those. We can detect those circulating tumour DNA or circulating free DNA, whichever way you want to call it, or circulating tumour cells, when a patient has fairly advanced disease. But when you're trying to detect it early, it's challenging, and you can't take 5 litre blood samples. So we're working on potentially using implantable devices to be able to do that. But we still really haven't understood, does this circulating tumour DNA reflect the particular cancer that we want to target, or a particular type of cancer that we want to target? What are the particular mutations? How do they reflect the tumour's mutations? Are they a particular subclone that circulates easily or lyses easily so you can detect it? So those studies need to be done, and need to be done fairly carefully. And we might get lucky in a couple of instances just jumping to circulating DNA. And we thought we'd be lucky, and we started like 3 years ago, jumped straight into it and said, oh, this is so cool, it's fantastic. And then basically we could only see it at very late disease, and it wasn't very helpful for us.
Yes.
thank you. It's a marvellous exposition of how the application of information technology particularly is going to impact on patient care. But I see another level of complexity arising here, because most pharmaceutical companies will have vaults full of drugs that cost a fortune to get to the final stage of patient trials, and they affected maybe 1 or 2% of patients, but the majority. Aren't they going to come forward and say, look, how about retesting all of these in your newly defined subgroups, and maybe we'll strike gold? How's this going to work out, I think?
So that's a very complex area. We've got patent life, etc. as part of that, so that it makes a challenge. And the regulators really drive this. So FDA made a decision maybe about 18 months or 2 years ago how they're going to deal with these small subgroups. And something they said was basically, if you, rather than doing a randomized control trial, etc., if you have 60 or 80 patients that have received this drug and you get a 20% or more partial response rate, which is a fairly low bar, we'll look at accelerated approval. And my phone started ringing. Basically, it gave it was an opportunity to start to move things forward. And now most pharma that I deal with will openly say that we don't understand the financial model in the future, because we always focused on giving drug for as many people as we could, at least in oncology, in a short period, a short window. But now we're finding that we can keep these cancers at bay for a long time, so it may be 100% of patients for 6 months, or it might be 6% of patients for 10 years. So they quite openly say, we don't understand that model, we don't know where this is going, but it's very sensitive to regulators. One thing, simply extending patent life would have a significant impact on drug development. To give the opportunity to use that biomarker-driven approach.
So, Andrew, how can you tell us a little bit about how, just following up on that question, how is the industry as a whole, the pharmaceutical industry as a whole, reacting to this kind of revolutionary? Because their original idea is this is a drug, that the trials working with that percentage of success, that's our product, that's what we're going to use. This is a completely different way of looking at things.
Yeah, so they are moving towards it. It depends on who you talk to. Pharma, in general, at least my interaction, is quite siloed, and you have the translational team is right all over this, but the individual drug teams are completely very different, very different strategy. They are responding. They've known for a while that a biomarker type approach is important, and we know that Roche developed a diagnostic arm, tried to buy Illumina, ended up buying PacBio. Other companies have diagnostic arms, etc., to be able to develop this, but they still don't. They're still driven by the regulators saying we need a pivotal randomized phase 3 trial to demonstrate this. And so how do you make that model work? Still finding their way, still trying to work that out. And it's an opportunity for us in the academic area to work closer with industry, understand what drives them, and find the right compromise that gives us what we want, gives them what they need to survive. And that's both a challenge but also an opportunity.
Right. Yes.
I remember hearing elsewhere that the sort of genetic changes in a specific patient's individual cancer can be quite diverse. I wonder if that's a misapprehension, and if it's not, how does that diversity impact turning this into useful clinical decisions?
I love it, the heterogeneity question. I always get the heterogeneity question. Good on you for that. And I've answered it before, so I'm prepared. How much genomics background do you have? Just okay. So
more than you think.
Yeah, that's it's the so this is a very benign question. I just want to ask you that you know it's coming at you. So think about cancer as an evolving organ. You're going to have your founder mutations, which are there right from the beginning, and they're present in all cells. And they're the things that we currently see most of. Then we see subclonal components. These are evolving in a different passage or a different way, and we can start to detect those in a certain allelic frequency. So this starts to become philosophy. So if you have a cancer that doesn't have a treatment, you need to drug your founders, those that exist in all cells. Otherwise, irrespective of what you drug somewhere else, they're going to continue. If you have other cancer types where you can initially, you have success with your drivers, knowing if you have a subclonal element beforehand is important. And we don't know the answer to that question. So an example is a particular therapy for lung cancer called erlotinib or Tarceva, and it targets a mutation called an EGFR mutation. The cancer is very clever. What it does, it delivers a mutation in that particular gene that stops the drug from binding. So it can't bind to that. And so you find another drug, it's an irreversible inhibitor, that bypasses that particular mutation. So what we don't know is if you hit the patient with both drugs up front, are you going to get a benefit longer term, or are you just going to get a really nasty resistant clone like you get in bacteriology or in microbiology? And so that really is the answer to your question, is that we don't actually know. I don't think that taking multiple biopsies from different areas and really studying the bits within the tumour at this point in time is vital. I mean, ideally what you want to do is take the whole thing out and mush it all out and then just do the whole thing on that. But that's not going to happen because you can't get every piece out. But the important element is that we can start to understand how that evolves and then think about how we treat patients differently. In some circumstances, a drug holiday might be the right thing to do. And we see some evidence in colon cancer where actually being off treatment for 2 months is beneficial. And so if you can imagine a situation where you have a dominant clone, and a dominant clone is dominant for a reason, so it's dominating the area. So if you hit that with your particular therapy, that decreases. And in a resistant clone, which is harder to kill, starts to grow up, you stop this drug and give it a bit of a holiday, so to say. They're sort of having their little, their battle there, and really understanding those dynamics, how you start to mold the cancer with your selective pressure. Because the minute you give them a drug, you're changing that pathway of evolution. And the cancers are very clever about becoming resistant. For example, a BRCA2 mutant cancer, it's defective because of a particular mutation, but it also makes them sensitive to a particular drug. So what this tumour does, it gets rid of the mutation that caused it in the first place, so it becomes resistant to that drug. So for some reason where we don't completely understand, but it's a really hyperacute evolutionary environment, and we don't know the mechanism as to why that happens. Did that answer it?
Anyone else? I'm going to ask a different kind of question. That this is the state of a developing understanding of cancer and a completely new way of thinking about treatment. What do you tell the students? What do you tell the medical students? How do you teach that?
Come into the lab. Yeah. I mean, I think it's, well, you need to know your basics, but I don't think it's as much as a leap forward as we might think it to be. Conceptually, it goes to a certain way, but if you asked me to sequence a cancer myself, I couldn't actually do it. If you asked me to code some analytical tools, I couldn't actually do it, but I know what needs to be done and what people need. I need to be able to do that. So it's like running an organization. You could be the CEO of Illumina, but you wouldn't even know how to, solder something. Right. Yeah. I didn't really answer your question, did I?
It sounds as though they live with a certain degree of uncertainty, but still.
Well, it's challenging. I think. I mean, it's basically education and understanding, and the opportunity of being in a lab, and also integrating scientific endeavour with clinical training for those people that want to do that.
Okay. Any more questions? I'm going to ask now for a vote of thanks to be presented by Professor Robert Steele, Bob Steele, as he's known to all of us.
Well, on behalf of everyone here tonight and on behalf of the Scottish Cancer Foundation, I'd just like to thank you, Andrew, for what was a truly superb lecture. I mean, as a surgeon myself, I'm very proud that it is a surgeon that's doing this type of work. And I'm not quite sure what it is, but some of the greatest advances in some of the most complex areas have been carried out by surgeons. And I think it's got something to do with motivation. And I was very interested to hear that this patient of yours with a very small pancreatic cancer who had disseminated disease within a few months really motivated you to work like this. And I myself have had similar motivations from my work. So what you have done is you have beautifully illustrated the heterogeneous complexity of cancer and how we really need the power of computing now to really understand it. And you've also illustrated well the shift from site-specific treatments to individual cancer-specific treatments. For many years, we have dreamed of the magic bullet that we can give to all cancer patients that will cure them. And we know that's not going to happen. But what we now can see is that the clear way forward is to understand the pathways that drive individual cancers and to target those specific pathways. And you've also illustrated very beautifully this shift away from these enormous trials of thousands of patients with blunderbuss treatment and looking for tiny statistical significant differences towards much more intelligent trials which are driven by the way the actual individual tumour behaves and grouping patients appropriately so that we can get answers much more quickly. A very beautiful illustration in our radical shift of thinking in this area. And thank you also for dealing with what were actually very complex questions in such a straightforward and lucid manner. And finally, I should like to thank, on behalf of the Cruden, behalf of the Scottish Cancer Foundation, thanks to the Cruden Foundation for their generous donation, which allows us to continue with these public lectures. Thank you, Andrew.